This presentation, Introduction to the Tools for Genomics-Assisted Breeding in Polyploids, is sponsored by the Tools for Genomics-Assisted Breeding in Polyploids Training Workshop and funded by NIFA, the National Institute of Food and Agriculture, within the USDA Specialty Crop Research Initiative Funding Program, and was held in January of 2021. Good morning, good afternoon, or good evening, depending on where you are. I appreciate you coming to this training workshop. I'm going to begin by giving you an overview of the project and some ideas of who's participating and what the challenges of polyploid crops are. So I'm going to talk about this in the following presentation. Now, I am David Byrne. I'm the coordinator of the project, professor at the Texas A&M University of Horticultural Sciences. I work in Rosa and prunus breeding and have done so for about 40 years. Now, the objective of this project are threefold, and we'll go through these one by one. So starting with objective one, develop the computational tools for genomic-assisted breeding of polyploid crops. And so in this objective, we're developing enhanced methods for genome association studies for linkage and QTL analysis, especially with multi-parent populations. And then better ways to assemble polyploid haplotypes from short reads. So we have those objectives that we'll be working with, and every year we'll be adding some more information and improved software packages to use for your analysis. Okay, your second objective is to validate the computational tools in public breeding programs. We're working with 6 different crops. We have potato and sweet potato. We have rose and ornamental. We have blackberry. We have turf grasses and kiwi fruit. So all by different programs. And we're going to be looking at different aspects of the software and comparing it side by side. So we're interested in allele dosage assignments, genome-wide association, linkage and QTL analysis, genomic selection, sequence-based haplotyping. So we'll be testing these on these various crops and various data sets, and also helping you do the same with your data sets, which comes to our final objective, which is to train. Could we have a beginning of all this software? We have some very nice packages that are being improved in real time with good documentation. And so we're developing the workshops and short courses to help you understand this and to use it better. And also developing a polyploid breeding community resource website that will be a depository for all this information. And again, if you were at the presentation and the workshop session yesterday, you would have seen how Slack channel is used to continue the inquiries and learning process throughout the year. So this is a training and outreach aspect. And for this particular workshop, we have over 300 people who have signed up and are participating. And so we have these 3 objectives, and they sort of work together. As we develop better tools, we continue validating, begin training, and then the people who are being trained in it are validating as well, and hopefully giving us input so we can improve the software as we go on. And so it's a interactive type process, and that's how I'd like to see it happening. But who's participating in this project? As I said, there's about 350 participants, but about equal amounts in the U.S. and international. And so in the U.S., we have 27 different states, people from 27 different states participating. And internationally, we have 32 different countries represented. So if you look at it by continent, South America is the one with the most. And this is really mainly Brazil, but also Peru. And then you look at Europe, you have Netherlands, Spain, France, and and England being the major countries represented there. In Africa, quite a few countries, and this has to do with the international research centers for the most part. We have people from Kenya, Cameroon, Nigeria, Uganda, and Tanzania. And then in Asia, Thailand and China, and finally New Zealand. Now, if you look at the participants with respect to where they're coming from, about 21% are from private companies. About 36% of the programs are public. And then fully 40 to 45, about 43% in this case, are students. This is both postdocs, graduate, and actually one undergraduate student as well. And this reflects the age range of our participants, very heavy in the 20s and 30s, that's the students and the postdocs, and then 40s, the young faculty, young breeders developing their programs. So again, we have a fairly young demographic, and these people are going to be shaping the future and how breeding's done in the next 10, 20 years. So it's exciting to see all this happening. You can see about 58% are breeders or breeding support. And this would include the molecular people, the biometrician, and all those types of people, the plant pathologists, entomologists, because breeders work with lots of different people. Of course, there's a small number of educators from the universities. They're also breeders and what have you. And again, students, about 45% in this tally are students. If you look at the participants' expertise, you can see breeding and genetics is the most common one. And in this, you could list various expertise for every person. So you have quite a few, more than 350 entries here. But again, computational genetics, molecular genetics are one of the most important ones. And then plant pathology, entomology, plant physiology. Again, breeding programs work with a wide range of people to make it work. But what about these polyploid crops? What kind of crops do we have represented? Now, this project is to benefit all crops, not just one or two. And a lot of the previous work has been done on one crop or two crops. This is trying to extend it to as many crops as possible that are polyploid. And so if you look at this, again, fruit, root and tuber crops are highly represented. In the fruit world, you're talking about blueberries, blackberries, bananas. I counted coffee in this because you're actually harvesting the fruit, kiwis and strawberries. And the root and tuber crop mainly is potato and sweet potato, but also some yams in there. Ornamentals, the major ones are rose and chrysanthemum. But a lot of the ornamental breeders work with multiple crops. And so there's actually a lot more represented that are not in this list. And then you have grass. Initially, I was thinking of turf grass, but we have forage grass breeders, and I included sugarcane in this as well. Vegetables, mainly legume crops, various cereals, and a couple forest tree species. So we have quite a wide representation with many different families and plant types. Now we look at the ploidy level. Tetraploid is the major one, with diploids as well. You may say, well, this is a polyploid project. But remember, when I asked the question, I asked, what ploidy levels do you work with? And many of these programs work on multiple ploidy levels. As rose breeder, I work on the diploid, triploid, and tetraploid level mainly. So I do work on various levels. But tetraploid is the one I'm concerned about. Of course, we have the hexaploids, octoploids, decaploids, and even higher ploid levels represented. But again, most commonly would be tetraploid and hexaploid among the polyploids. And triploids are extremely important, especially bananas being a major triploid crop. And quite a few of the ornamentals have triploid components as well. You look at the size of the breeding programs, you can see that 51% have breeding programs with 5,000 or less seedlings, about 26% between 5 and 20,000, and about 23% above that. So again, a wide range of program sizes as well. So we have various groups of crops, and from this list you can add the forest trees as well. The ploidy levels, again, range from triploid all the way to decaploid and higher. The genome size is from about 300 to 36,000. Of course, this is mainly you're seeing these larger sizes among the grasses and things like that. And of course, you range from allo to segmental to autopolyploid. Now, we're mainly concerned about the more autopolyploid situation here. And then if you look at the breeding characteristics of these crops, it's very common to see interspecific interploidy crosses and hybrids. The plants generally are medium to large size. Most are perennial, not all of them, but most of them are. And you have quite a few clonally propagated crops. They tend to be outcrossing, exhibit some inbreeding depression, not all of them, but it's not uncommon. Self-incompatibility and interspecific incompatibility issues. So you're running against a very heterozygous polyploid crop. Breeding cycles anywhere from 1 to 4 years for most of them. Some might be even longer. And again, you're seeing a lot of interspecific crosses, and then the common field approaches to breeding, recurrent selection, backcrossing, and pedigree approaches are taken. So a lot of different, really quite diverse, but all tied together by a lot of heterozygosity and higher ploidy levels. But if you ask the participants whether they use molecular markers in parental seedling or genomic selection, this is what you see. The most common response was sometimes or never. And so we're just at the beginning of using some of these techniques in some of these crops. And so we got a long ways to go, but I think it's really exciting, particularly because when you look at the impediments, one of the major impediments are software tools and analytical expertise is what we're aimed at. Now, of course, one big impediment, just a lack of markers for traits of interest, especially for seedling and parental selection. But again, without the software tools and analytical expertise, that's hard to get. And of course, other things that are of interest, of course, phenotyping tools, genome sequences, etc. And then if you look at how many of these participants have actually used the software, fully 90% have not. So this is really new territory to most of these people. They're very brave to be here and to start learning. So we have a lot of interest and a lot of confidence that's going to make a difference. And that's exciting. And so this, you can see medium to high confidence that within 10 years, it's going to have a significant effect on a breeding program. So either from parental selection, seedling selection, or genomic selection. All of them, there's a really high confidence that it's going to make a difference. So what are we going to do in this first training workshop? Well, yesterday, if you joined us at the Computational Support Group, these 5 young scientists helped the group install their software and answer questions and helped explain what a Slack channel is and how to use it. And we had about 160 participants in that workshop. Now today, of course, I'm giving you the introduction. After me will follow Dory Main, who will introduce you to the Polyploid Community Research website, which is under construction, but she's rapidly putting new material there. And once we get done with this workshop, all those materials will go on the website as well. And so after that, we'll talk about genotype calls, and Roland will give an overview. Marcelo will talk about supermassa, Lindsay, polyrad, and Roland will follow up with fit poly. So 3 different approaches to calling dosage in polyploid crop data. And then after that, we'll have breakout Zoom sessions. So in the email I sent you, you have all the Zoom links for those sessions as well. And then on January 14th, we'll go to linkage maps and haplotype reconstruction. And again, we'll have an overview. And then we'll talk about 3 sets of software. Peter Bork will talk about PolyMapR. Marcelo will talk about MapPoly. And Chao Z and Jeff will talk about PolyOrigin. So they'll talk and then give a demonstration. And again, breakout rooms as we had before. And then the final day on January 15th, on Friday, again, we'll talk about quantitative trait loci analysis. Here we'll have Chris give an overview. And then Peter talk about PolyQR or QTLR. G will talk about QTL Poly. And Jeff will talk about DIAQTL and GWAS Poly. And then again, follow with questions and answers. Now, what are the expectations? What do I expect you? What do we expect you to come away with this? Well, again, we're going to give you a basic theory and an introduction to the software and a quick demonstration. It's not enough to learn it, but we have a way to ask questions, and you can ask questions while the person is presenting. There'll be somebody monitoring the chat, and hopefully they'll be able to answer your question. And if they can't, they'll interrupt the presentation and ask the presenter to talk to you. And so what we hope to do is that you get to the point where you have confidence to really dig into this and start using it, because really the best way to learn these types of things is by doing. And additionally, hopefully develop a good network of collaborators as we go forward. This is a 4-year project, so we'll have 3 more training workshops like this and perhaps other things in between as we need. And so those are things that hopefully we'll develop. To follow up, we'll have recorded talks posted on the website. You can have your questions via Slack channel. If we need other things as we find out, as we work through this, we'll see what we can do. I will be sending out surveys always to get your feedback. I want to know what's working, what's not working, what features you may need in the software that we're not addressing, clarifications needed in manuals, suggestions for improvements, whatever you have. I'll be sending surveys to get your input. So please respond to all surveys. This presentation has been sponsored by the Tools for Genomics Assisted Breeding in Polyploids Research Project. For more information, you can email us at toolsforpolyploids.tamu.edu or visit our website at www.polyploids.org. This project is funded by NIFA within the USDA Specialty Crop Research Initiative funding program.